Hi, and welcome to the Neurological Foundation's Discovery Sessions. The Neurological Foundation has been funding research into neurological conditions and disease for nearly 50 years. We have funded some amazing scientists and clinicians throughout their career who have gone on to conduct groundbreaking work and discover fascinating things about the human brain. We hope that these digital discovery sessions will help all our followers, supporters, and fellow science enthusiasts learn new things about one of the most complex organs humans have, their brain. Today's speaker is Akshata Anchan, a doctoral candidate from the Center for Brain Research at the University of Auckland. Akshata was a recipient of the Foundation's Gillespie Scholarship and has investigated how cancer cells can get through the blood-brain barrier that surrounds the brain. We are grateful to Akshata for taking time today to speak to us about her exciting work on the blood-brain barrier and cancer cells and teaching us all about this unique part of the brain.
Thank you for coming along to this event today, hosted by the Neurological Foundation. And as you know, the topic for today is, of course, is titled An Unwelcome Visitor to the Brain. And that's essentially referring to how cancer cells enter the brain, where the cancer cells are the unwelcome visitor. So I was going to spend a second saying hello, but Jackie gave me an extremely detailed introduction. So as you know, I'm a PhD candidate working at the Center for Brain Research at the University of Auckland. And I'm able to do that thanks to the Neurological Foundation Gillespie Scholarship, which essentially funded my PhD. And my project is essentially looking at bringing cancer and brain disease together. And it's under my supervisor called Dr. Scott Gray. And this is just a cute little picture of our lab group. But moving on, today's talk, the focus point for today's talk is how does melanoma, which is a skin cancer, get inside the brain? There is another part for cancer surviving in the brain, which is detailed here. However, for today, we're probably just going to focus on the first part, which is how it actually gets inside the brain in the first place. So first of all, why melanoma? Of all of the cancers. And this is because, well, melanoma has the highest incidence in New Zealand compared to anywhere else in the rest of the world. Australia comes a close second, and the reason for this is actually for several different factors, predominantly because of the geographical location that we're in, also because of the lack of air pollution in New Zealand, and also, of course, as we know, because of the hole in the ozone layer. So that's why melanoma has an extremely high occurrence rate in New Zealand. But what's very interesting is that melanoma also has high propensity to migrate specifically to the brain. Now, what this means is that while melanoma, which is a cancer that occurs at the level of the skin, can migrate to lots of different parts of the body, including the lungs or the liver, it seems to preferentially migrate to the brain. And unfortunately, when patients get melanoma, and if they have a poor prognosis, this poor prognosis is actually closely linked to the ability of melanoma to be able to invade into the brain. Sorry, I hear that you cannot see my presentation, so I'm just going to see if I can fix this. Awesome, cool. So don't worry, you haven't missed a lot in terms of the presentation. It's just been words up until now. But essentially, as I said, melanoma has extremely high incidence to occur in New Zealand, and it has propensity to metastasize to the brain. Also, it is the propensity of melanoma cells to metastasize to the brain that leads to the poor prognosis. And so as we go forward, how does this actually happen? Well, as we know, if we spend too much time out in the sun without a lot of protection, sometimes we can get some moles occur onto our skin. And that usually happens due to the UV radiation produced by the sun. And typically, this is actually not an issue. The moles are usually benign, and they don't really hurt us very much. However, sometimes the UV radiation can cause mutations to occur in these little moles, or as we call them, melanocytes on the skin, which are the pigment producing cells of the skin. And that causes these cells to start growing and proliferating, as shown over here, and then they can become a tumor at the skin. Now, if this tumor is particularly invasive, then it can start invading into the profound layers of the skin and then find its way into a blood vessel, which might be in the profound layers of the skin. Now, once a melanoma cell can go into the blood vessel, it essentially has access to the circulation. And this means that it has access to all the different parts of the body. And that's how cancers, including melanoma, get to different parts of the body. Now, once it's in the circulation, it can go anywhere at once and then go out of the blood vessel, as shown in this picture here, to a secondary site. And as we said earlier, melanoma has a preferential migration site to the brain. So that means over here, it's coming out of the blood vessel at the level of the brain. So as we said, a melanoma cell at the skin can go into the blood vessel, travel to different parts of the body, and to get into the brain, it would have to get out of the blood vessel at the level of the brain. And this is essentially the main first part about melanoma migration to the brain, which is how exactly it gets inside the brain at the level of the blood vessel. And there's another part to this, which is how it survives once it gets in the brain. But for today, I'm going to focus on the part one, because that's essentially what my PhD has been focusing on. However, to get into the brain, this should actually be quite difficult for melanoma cells because of the presence of what we call the blood-brain barrier. Now, we're just going to go to a picture of the brain so that we can look at this a little bit more in detail and spend a little time on this slide just talking about what exactly the blood-brain barrier is. Now, as we know, the brain is an extremely energy-demanding organ. It has a lot of activity occurring, which means that it needs a lot of oxygen and nutrients. And that's why it's completely surrounded by these blood vessels to give it a good input of blood flow, which can then provide the brain with all the nutrients and oxygen and energy that it needs. So if we look at maybe this blood vessel here and look at a cross section of this blood vessel so that we're looking at the blood vessel head on, essentially if you cut the blood vessel in half, this is kind of what it looks like at the level of the brain, where this is the blood vessel, the blood flow would be in here, and the blood vessel is surrounded by the cell that makes the blood vessel wall. There's also other cell types of various shapes and sizes that support this blood vessel wall. And together they make up what we call the neurovascular unit, which essentially form the blood-brain barrier. And these exist at the level of the brain only to protect the brain from any blood-borne pathogens or injurious substances such as bacteria or viruses that might be in the blood vessel over here. And that can happen quite easily because if you imagine that you have like a cut or something that gets a little bit infected, then you can have a lot of pathogens in your circulation. And as I said earlier, then they'd have access to all different parts of the body. And we don't really want anything that's injurious in the blood to get into the brain, because if that happens and if the brain gets injured, then that's obviously not going to be very good. So that's why there's this blood-brain barrier that essentially protects the brain from anything that's bad in the blood. And this specific mechanism is extremely important for the brain and is actually quite specific to the brain. There's nothing like this in any other part of the body. The blood vessels and other parts of the body don't have this barrier. And that's particularly a specialization of the brain to protect it. So I'm actually just going to go back quickly because I want to talk a little bit more about this black bit here. So as you can imagine, for anything that's in the blood, including viruses or pathogens, or for say a melanoma cell, to get into the brain, the first sort of cell type that it would come into contact with is this cell type, which makes the lining of the blood vessel wall. And that means to get into the brain, it would have to essentially, well, cut through it. However, the specialization of this blood vessel wall is that it's adjoint to each other. So the the vessel is lined with a lot of these cells, where the cells are attached to each other's very, very, very tightly by what we call tight junctions, which is essentially what this black box over here is trying to represent. So if we zoom into that black box, say this bit here, which is the cream-colored boxes, are two cells that line the blood vessel, and they're adjoined to each other. They're identical almost, and they're joined to each other by all these proteins that essentially attach the two cell types together extremely tightly, making a physical barrier against anything that's in the blood. And this means that if a melanoma cell were to get from the blood, which is here, into the brain, which is here, it would have to traverse through all these tight junctions and break to the barrier to get through. So how exactly does it do this? And what do the melanoma cells have that allow them to interact with and then break through what we call the blood-brain barrier, or rather the cells that make up the blood vessel wall of the blood-brain barrier, which are essentially the first cell type that the circulating melanoma cells would come into contact with. So now I'm going to talk about what I found in my first year of my PhD. And what I found is that, unfortunately, melanoma cells are negatively able to affect the brain blood vessel cells. And I've learned this by adding human melanoma cells, which were resected from a patient who kindly donated their tumor to us, to some of the cells that form the blood vessels in the human brain. And I found that adding these to the blood vessel cells decrease that barrier integrity of those cells. Where, say, if we use these cream-colored boxes as a representation of the cells that make the blood vessel wall, when I add the melanoma cells to these, they essentially rip through those tight junctions, as I pointed out earlier, those big boxes, they rip through them to then traverse through. So I'm going to talk a little bit about how exactly I discovered this, and it's a little bit complicated, but this is essentially what it looks like. So it's called ESIS. It actually stands for Electrical Cell Substrate Impedance Sensing, and essentially a machine that does a lot of the measuring for us. It's got this computer digital system, which essentially displays the data, which is what we can interpret. It's got this big machine at the bottom, which does all of the sign C measuring. And it's got this apparatus on the side, which is essentially our interactive path for us to be able to put our cells into. And as I said, we just call it ESIS for short. So that apparatus that I pointed out earlier, if we zoom into that, essentially over there, we've got this big plastic plate. It's just made of plastic, and it's a big rectangle. And in there, there's all these little tiny wells, as you can see over here, that's zoomed out over here into this view. And the wells allow us to put live cells in there. They essentially act as compartments that we can put the live cells into. The wells are also lined with these gold electrodes at the bottom that you can see sort of interdigiting into each other. And so the electrodes essentially mean that we've got current in the system. We've got an electrical system, and we can use this current to look at cell behavior. And it's really quite neat how it works. Because then once we've got this current, we can analyze any changes in the current readout that we get in our digital system, and we can interpret those changes in current to understand if there is any changes occurring in cellular behavior. So the current actually changes. The current that's in the well changes according to whatever is inside the well. So what happens is, if we've got a well that's completely empty, the current in the well essentially is a flat line because there's nothing that's happening in the well. Now, if we take some of our cells that line those blood vessels in the brain and we add them into the well, what we see is that the current goes down. It decreases. And that's because these cells, they've got physical mass, and they act as an impedance or as a resistance to the path of current flow. So less current can physically flow through these cells, which means that the current drops. However, if we take some melanoma cells and then we add these on top of the cells that make up the blood vessel walls, then the melanoma cells start essentially poking holes all through these cells, all through the cells of the brain. And that means that those tight junctions are broken, which means that there's less resistance, so the current can flow through again and the current goes up. And we can interpret all of this data to understand how melanoma cells disrupt the barrier integrity of the blood-brain barrier. Because we're using current, which is an electrical system, it's extremely fast and extremely sensitive because it happens really in the matter of under a second. So we can essentially look at things that are happening under a second, and we get information on cellular behavior almost as soon as they occur. And that's actually quite unique to the system. There aren't a lot of systems out there that allow us to look at cellular occurrences so fast. And furthermore, we can actually detect really small changes that are occurring as well. And remember, this is important because cells are obviously really small and you can't actually see them by eye. So what I found in the first year of my PhD is that ESIS allows us to look at the structural and temporal changes of these cells, where we find that when we add melanoma cells to the cells that line the blood vessel of the brain, they disrupt this side part here first, where they're disrupting these tight junctions, and then they intercalate through the endothelial, through the brain cells, and then go inside and start spreading out underneath, which kind of makes sense if you think of an invading melanoma cell. Okay, well, that's all great. And we get a lot of nice graphs out of it, but that looks a little bit boring. Can we actually see these cells and see exactly what's happening with these cells? And yes, we can. So over here, what I've got is the same cells that I've been talking about before, which are the cells that line the blood vessel at the level of the brain. And I've essentially stained them green so that we can actually physically see them. We can also look into this circular area, which is kind of the focus point for this image, where before I add any melanoma cells to them, they look quite happy. They're attached to each other. That's one cell there. That's another cell there. That's another cell there. That's another cell there. And they're all attached to each other quite happily, forming that barrier. And again, they're quite nice and happy. And then I add my melanoma cells in. So these big circular cells with the giant halo around them, they're the melanoma cells on top of the blood vessel cells. And what we see is that over time, the melanoma cells start to become darker. And what that's showing is that the melanoma cells are actually going into the layer where the blood vessel cells are. And you can see over time, they start to spread out and their little processes start coming out like so, because they've integrated and migrated into the cells that make up the blood vessel. And you can see even over here on this side that they've really made this new place their home, essentially. And this is another view showing the same thing, where on the top panel, I've got the melanoma cells before addition. I've got the blood vessel cells before addition, and then the second one, where I've got the melanoma cells added. And if I look at just the cells that make up the blood vessel cells, without the melanoma cells, again, they look really nicely attached to each other. However, with the melanoma cells, you can see all these holes have occurred in that layer, in that nice, pretty layer of the cells that form the blood vessel cells. And essentially, this is how we think the melanoma cells are going through, is by just disrupting that barrier and getting into the brain. So the next question is, how exactly do the melanoma cells do this? They shouldn't be able to, but they do anyway. And during my PhD, I've looked at a range of different things that could help melanoma cells insert between the brain blood vessel cells. And these include the proteins that could be expressed on the surface of the melanoma cell, like so, which could then go and interact with the proteins that make up the tight junctions, or it could be proteins that are secreted by the melanoma cells that could also disrupt the tight junctions, like so. And now this is a really busy table, but essentially what it's showing is that I've looked at a lot of different proteins on their own and more. However, they're not really doing anything on their own. So I'll move. This is just to show that I've looked at a lot of things. But if I move on from here, what we found is that looking at all of those different proteins individually doesn't really seem to affect the blood-brain barrier. And so we wondered perhaps multiple things are occurring at once to then disrupt that barrier. So now we're looking at proteins in combination, and we believe that melanoma cells may actually utilize several redundant mechanisms to allow their interaction at the blood-brain barrier. And I'm not going to get too technical, so I'll just leave it at this sort of vague term. But what we're finding is that, yeah, melanoma cells might be using different things together to do so. And this actually makes a lot of sense because, as I mentioned earlier, the blood-brain barrier is here to protect our brain. It's extremely specialized in doing so, which means that it's probably got a lot of methods to keep pathogens and melanoma cells out. And that means that to get through, melanoma cells must also adapt and become specialized to then break through the blood-brain barrier. So now what we're trying to do is essentially screen the melanoma cells for everything that they could possibly have that is of importance in literature. And we want to do this at the level of the RNA. So we're looking at the nucleic acid level to then be able to figure out exactly what it is, just anything that the melanoma cells could be expressing, and that also expressing together to get through the blood-brain barrier. And then once we know what exists, we can then try to determine how important it is and how crucial it is in aiding the melanoma. But as I said earlier, that was only part one, and then there's a whole other part to this where we look into how the melanoma cells actually survive once they're in the brain, and that'll form part of a future project. As I mentioned earlier, this is just explaining what the part two is. And so we're doing this, we're looking at all these proteins and we're trying to identify a whole range of these proteins because we're considering that they might actually be different for different people. Because a melanoma is it's a cancer of an individual, you'd expect different people to maybe have different proteins that are expressed by the melanoma cell that aid their traversal into the brain. And for the future sort of my research, I want to identify if this is actually true, and then also consider what that might mean for therapy. Because we need to realize that people are different and they're affected differently by melanoma and also other cancers. And so the treatment might actually work in a very similar manner in that we need to understand what kind of proteins are actually expressed before we're able to get rid of it, especially because therapy can actually be quite harsh on people. And if you treat someone with the wrong thing, then they might have to suffer through the side effects, but not actually see any benefit come out of it. So we need to really do a lot of research around this topic and understand what kind of proteins are expressed and if they're actually different amongst different melanomas, which come from different people. And we see evidence of that happening. So we want to be able to screen for all of this. So we know what's available, we know what's there, and then once we know what's there, we can try to figure out how to get rid of it. And yeah, it's not just what's likely to work before actually using it. Therapy is also very expensive, not to mention. So what can we do? Well, as a scientist, I can keep conducting research and work together with other scientists around the world to help find more answers. The more collaborative work we do, the more information we have about what kind of proteins are expressed, the faster we can progress the drug discovery that comes out of it. Especially because not everyone is an expert at everything. Everyone's got their specialties in different areas of melanoma migration, and we need to be able to adapt to all of that. And essentially, we're trying to aid scientific research to cover a lot of what's unknown. And what we find as we do more and more of this work is that there is quite a lot that's still unknown. And just to finish up, that's essentially what we're trying to do anyway in our lab. We're trying to collaborate with a lot of people to be able to understand exactly what's happening. So as you know, I've got my supervisor, Scott Graham, but we're also working with immunologists and anatomists. We're working with the Auckland Cancer Society Research Center, pharmacologists, and several other people. including the Neurological Foundation Human Brain Bank, which is actually an invaluable resource that we have in New Zealand that allows us to look at brain tissues that have been very kindly donated by patients with a particular disease. And we can look at these and we can investigate these to exactly understand the differences between them. And then, of course, the Neurological Foundation for funding the project altogether. And what's extremely important is being able to make these collaborations internationally as well. As I said, now melanoma is quite specifically specifically difficult in New Zealand, yes, but it does occur in all parts of the world, and so do other cancers. So it's really important to be able to communicate the research that we're doing in New Zealand with people around the world, and see if there's any differences or any similarities that we can cross correlate. So that's a really big part of it, and I've been trying to do that. Again, this was funded by the Neurological Foundation during my PhD, which allowed me to take a lot of my work to an international conference in Europe, where I could communicate some of the research that I was doing in New Zealand with clinicians in Europe, which was really, really quite cool, because they're essentially working with the patients face to face, whereas I'm doing more lab work. So it was really interesting for me to learn the differences and variabilities in these melanomas that they see in their patients. But it was also really cool for them to see how we do the mechanistic stuff in the labs. And that's essentially the idea over here, is to just keep doing more research so that we can better understand what's happening with these cancers. And the better we understand it, the more likely we are to be able to stop it. And on that note, I will thank you for listening. And just another acknowledgements to the supervisors and advisors who've helped me through my PhD, other lab students who obviously share the same work, other members of the CBR at the University of Auckland, which is the Center for Brain Research, but most importantly, to thank the donors and their families, and also the funders who make this all possible. So thank you for listening, and I think we'll take questions now. Our first question is, do we have high rates of brain cancer since we have high rates of melanoma in New Zealand? So when we consider rates, we're actually thinking of them in the population base. So we're thinking of how many cases we've got, say, per 100,000 people. And if we look at that, then if we're relating to melanoma, then yes, brain cancers that happen in because of a melanoma. So it's a secondary brain cancer, in that the melanoma occurred somewhere in the body and then it went inside the brain. Those rates are higher also in New Zealand, and that's linked to the fact that we've got more melanoma itself. Brain cancer as a whole is a totally different topic, because there are some brain cancers that occur just in the brain, but that's a little different, and then the rates for that vary across the world. We've got another question. Is there hope for being able to turn off the melanoma cells impact in the brain? Yes, there is. There is actually a lot of hope. And that is referring to that part 2 that I was talking about, which is looking into how melanoma cells survive once they're in the brain. Now, one thing that I didn't mention in my talk was that we've actually got a lot of drugs that have come out of some of this research. Not me specifically, but just people doing this kind of research around the world. We've got drugs where we're essentially trying to boost our immune system, which is one of the only things that can actually adapt as fast as melanoma can, and also as other cancers can. And we're essentially trying to boost the immune system to take over and kill the melanoma cell even once it's in the brain. And what we're finding is that in clinical trials and in patients, it's actually working for some patients. It doesn't do it for all of them, which is why we need to figure out why not. But it seems to work for some patients, where once we boost their immune system, it actually gets rid of the melanoma quite effectively, because the immune system just kicks into place and it just does what it's there to do. So there is some hope. We're already seeing it work. It just seems to work for some people when it works, it's really, really good. But sometimes it doesn't. And we're trying to figure out now why exactly that is. And we think it actually might be because some people express some proteins, some people express other proteins. And it's just figuring out what exactly we need to target, which seems to be the difference. Cool. Another question. Are the melanoma cell donations used in your research from a single donor, a few, or many? That's a good question. They're from many. They're from many, many donors. So I've used lots of different melanoma lines, which have been essentially multiple different patients who maybe had a tumor and maybe had a tumor that so that's metastasized to a different part of the body, and because it was hurting them, someone decided to take it to the hospital, and the hospital took it out for them, and then they asked if they're happy to lend this tumor for research, and the patient very kindly said yes. So then we take the tumor and then we essentially just chop it up and we make it into a cell line that we can use in the labs to assess. And that's essentially, and it comes from many different patients. Cool. Sorry, I'm just reading another question. If the proteins differ in different people, is there a risk for your research that your samples might not have all the relevant proteins? Well, that might be partially true, but I don't think so, because we'd like to think that some of the main, although there are a lot of different proteins between the different people, we'd like to think that there are still some key things that we can key in on once we've screened for everything. I think it's more about just knowing that we've screened for everything that we know of in literature, rather than that there might actually be something that's missing altogether. We know of a lot of proteins that are already involved in these pathways. It's just that there's a lot of them. Like the table that you saw in the presentation, that's a very, very conservative table. When you look into the science, there's actually so many different things that we could be looking at that we know of. So we're essentially trying to key in on those and scan across a large range of different melanomas from a lot of different people to see if some key hotspot proteins essentially start spiking. So that's what we're going to go for. Cool. Another question. Do you know of any pathogens that have similar burrowing behaviors? And could the melanoma cells be using something like that? Yeah, so we do actually. And a lot of the research that comes into looking at melanoma getting into the brain actually stems from a slightly different disease. I'm not sure if you're familiar with multiple sclerosis, but this is an autoimmune disease, which essentially explains that the immune system of the body becomes a little bit hyperactive, and it starts targeting the brain. And essentially what happens is immune cells that are in your blood get into the brain through a similar mechanism where they're essentially breaking through those tight junction barriers at the blood-brain barrier. And we're actually thinking that the melanoma cells are doing something very, very similar. And I've seen evidence of that in my studies, where, as I say, we see some of those hotspot proteins coming up in melanoma cells that we've also seen in the past coming up on immune cells. So yeah, there is some potential for there to be some crossplay between the mechanisms between different diseases. All right, where am I? Sorry, there's a lot of questions. It's really good. So techniques to pass through cell layers. I'm not entirely sure what that question means. I assume it's asking if there are lots of different techniques to pass through the cell layers. I'll assume that's what it's asking. Or we'll just go to question 5. Okay, so I'm not entirely sure what that question's asking, so we're just going to skip it for now. But there are... Okay, cool. Now the next question is, there are many types of tumors that can form in the brain. Are melanoma cells the cause of all of them? No, they're not. No, they're not. Lots of different types of tumors from elsewhere in the body can form brain tumors. So while I said that melanoma cells have the highest propensity to metastasize to the brain, what we find is that in New Zealand and also in other parts of the world, one of the most likely cancers to go into the brain is actually is actually lung cancer and then breast cancer. And we found that the three most common cancers that is found in the brain that come from a different part of the body are those three, which is lung, breast, and melanoma. There's some other cancers that go into the brain as well, but those three are the top. And then there's also cancers that happen in the brain. So there's glioblastoma multiforme and other cancers that form from cell types that are resident to the brain. So those are called primary brain tumors because they occur in the brain, whereas I'm looking at cancers that occur in another part of the body and then go into the brain. So no, there's lots of different types of cancers that can go in the brain. And there's a second part to that question, which is, or are there cells that can also break the barrier? Yeah, so all of those different cell types would be able to break that barrier to get through. There's some differences in how they do it. We think that melanoma cells and also lung cancer cells potentially break through the barrier, but there's also some evidence showing that some cells might actually just go through the cell body of the blood vessel wall, just go through, just essentially just rupture through and get through. But we don't exactly know how that happens, which is why we're doing the is, does the differences in the vascular network construction by individual come into this, such as connective tissue disorders? Okay, there is, and this, I think this is going to be the last question for now. Yes, I'm not entirely sure if connective tissue disorders are the only example of this, but we do expect that if there is essentially a weak spot in the blood-brain barrier, or if there's anything at the level of the brain and the blood vessels in the brain that can sort of be used as a wounded spot that these melanoma cells or other cancers could target, that might mean that the melanoma cells are more likely to get through. What we expect, however, is that your brain and the systems in your brain are able to pick up on that and make up for that, and that's what usually happens. It's just usually a combination of a lot of things going wrong that we don't really notice because we can't see it happening, and then that's what usually leads to the migration that ends up occurring. But yeah, like if there is a weak spot, it'll make it more likely, but we hope that that's not the only thing. There's a lot of different things that need to occur because migration, any kind of migration, not only to the brain but anywhere, is actually an extremely inefficient process. Your body's built to protect itself. It's just when a lot of things go wrong back to back when you see these things happen. And the biggest thing you can do at this point, especially for melanoma, is if you see a melanoma spot come up there, or if you see a mole that looks a little bit dodgy, then you go get it checked up as soon as you can, because melanoma, if it's at the level of the skin and if it's detected, then your doctor will actually just get rid of it through surgery. And most people are completely cured of melanoma before it progresses too fast, if it's picked up fast enough. It's just once it starts spreading to different parts of the body, which makes it difficult. But I think that's all for now. For the questions as well. So thank you for coming along to the talk. Sorry about the hiccup at the start with the presentation, but I hope that we learned something today. And thank you for the Neurological Foundation for hosting us. But I think we're done.